Yeah, hi, it's Dr. Untitled again. You know me from my previous tapes. I hope you've enjoyed those. As I mentioned before, I've been involved in research for over 45 years of scientific research. I got my PhD from the UCLA School of Engineering and Applied Science. And I want to read you something that Darwin wrote to a friend of his back in 1871. He says here, but if, he wrote this to his friend Joseph Hooker in 1871. He says here, but if we could conceive of some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc. present, that a protein compound was chemically formed, ready to undergo still more complex changes. You know, you have to realize, Darwin lived 150 years ago. He had no notion, he had absolutely no notion of how complex life really is. When he looked under a microscope, he saw a blob that was a cell. He had no idea of the molecular foundation that blob is based on. So basically here, Darwin wants to go one step further. In his original book, he proposed that life would start from some single cell. So he doesn't know how that cell got there. But here we're talking about something totally different. Life starting from chemicals, chemical evolution. Is that really possible? That's what we're going to look at today. And there are people going around today who have all this knowledge. These are very brilliant people who believe that this is even possible. Let's analyze it a little closer. So we have Darwin's warm little pond based on the letter that he sent Joseph Hooker in 1871. We have a simple cell here. First of all, back in 1952, there were a couple of scientists, I think in Chicago, that actually wanted to see what happens when you combine what they thought was the primordial atmospheric Earth. They took water, methane, this is Miller-Urey, water, methane, ammonia, and hydrogen, put it in a glass container, started shooting some electrical sparks to there that simulate the primordial atmosphere, and then when they finished, they opened up this container and analyzed what was inside, and they found that they formed amino acids, the basic building block of proteins. Amazing! They got very excited about this. The only problem was, see, in life, proteins are composed of amino acids. In life, 20 amino acids are used. But for inorganic, non-biological systems, amino acids are either left-handed or right-handed. So if you have 20 left-handed ones which are used in life today for synthesizing proteins, you also have the right-handed ones. So you don't have only 20, you have 40 individual amino acids there. So this complicates the situation with going from chemicals to life. You don't only have 20. And we don't really know why in nature we only have 20 left-handed proteins, left-handed amino acids that are used to synthesize proteins. We don't know, but based on their experiment, you get both. By the way, there have been studies done after Miller-Urey who indicated that it's quite possible that the atmosphere that they proposed for the primordial Earth never existed. So this may never even happen on Earth. But let's assume that we have all the amino acids under the right conditions on Earth, and all the nucleic acids that we want on Earth through these different processes. We're just going to make that assumption, even though it's unrealistic. Can we still get life from chemicals? Well, let's take a look at the Minimum Genome Project. NASA started this back thinking about this back in the in the 60s, and it's a project that's continued on until, I think it's even exists today, at least into the 2000s, 40 years or more. They wanted to determine what was the minimum DNA size, the minimum bacteria that could survive with a minimum number of genes for producing proteins, because proteins are the basis for life. So what they did is they first of all looked at the simplest types of bacteria. And the simplest one turns out to be genitalium. It's a bacteria that actually causes human infection. They started off with 482 coding genes to produce these proteins. And they knocked off one gene at a time. 1, 2, and 3. They went down from 42 to 3D2, and when they went down just 1 or 2 more, it ceased to function. Life ceased to exist in the simple cell if you have less than 382 proteins in there. You can't, even if you form 1 or 2 proteins, it's not going to give you life. You need 300, based on our best knowledge today, based on all this research that was done. You need, at least for the most basic life form, you need 382 proteins and the coding in the DNA to produce those proteins. What about the chicken and the egg situation? What came first? Was it DNA or was it proteins? If the DNA came first, then the DNA could be coded to make proteins, right? And what if the proteins came first? Well, it turns out that proteins themselves are used in the DNA. There's a protein called RNA polymerase, which, you know, the DNA basically codes for protein. Each gene codes for a different protein. There's a protein that opens up the double helix of this simple bacteria, reads it, and forms an mRNA, which is then used by the ribosome to manufacture proteins. The proteins are the building blocks of the cell. If you didn't have this RNA polymerase protein, you couldn't read the DNA. The DNA would be totally worthless. So you need proteins, and you also need the DNA, the coding. You need both. Proteins are also used after the ribosome makes the string of amino acids that's a protein. That thing is totally useless. It has to be folded into an exact 3-dimensional shape, and there are proteins called chaperones that do this. So you need both proteins and coding from DNA to have life. You can't have either one and not the other. What about, let's assume in your wildest imagination you were able to form a bunch of, by accident, according to Darwin, you were able to form some proteins. We're going to get into the odds of what it would take to form a protein, but let's assume for argument's sake that you were able to form these. You formed 10 in South America, and 7 in North America, and in the primordial Earth, and 17 in Europe. Will that give you anything? Of course not. Because you have location, location, location. These proteins, this DNA, can't just be anywhere. It has to be localized into the smallest area of this genitalium. It's under the cell membrane. That's why the cell membrane is so important. You need a cell membrane. Otherwise, the proteins, the DNA, everything will just flush all over the place. You need a drop of liquid has 50 million bacteria in there. You need this localized. You can't just have proteins floating around anywhere and amino acids floating around anywhere. So it's not only that you're synthesizing the proteins called by accident, randomly, by chance, but they have to be together. You need a location, 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 like in real estate. And then for life, you need replication. What makes life different from anything else in the universe? It's able to replicate. It's able to reproduce. It's a very complicated process. First of all, in these bacteria you have a ring DNA. It's a closed ring. This ring has to double itself. It has to reproduce. And then the cell has to split into two cells. You know how complicated a process that is? We don't really understand the whole process today, even with all our knowledge. So how do you get replication? You can have all the proteins and DNA you want, even localized. Without the replication, without the reproduction, you have nothing. You don't have life. Life means division of cells. Furthermore, as we mentioned, the DNA ring codes for, and there's a protein that makes the mRNA molecule, which then floats over to the ribosome and goes into the ribosome. Ribosome is your cell's factory for making proteins. It reads the mRNA code and produces the string of amino acids, which is the protein, and then it still has to be folded, of course. Do you know how complicated ribosome is? Ribosome is a combination of nucleic material and protein material. It's made out of RNA, complicated RNA, with proteins, 30 proteins or more, joined together. It's a most complicated structure next to DNA in the cell. How do you form that first ribosome in there? If you don't have ribosome, you don't have proteins. Even if you accidentally make a protein or two, it doesn't give you life. You need an effective way to keep on producing these proteins, because proteins have a limited lifetime. They get denatured. If they're not inside the cell, can you imagine you form this protein and somewhere in the ocean, they're affected by the pH, the acidity. They're affected by salt, like in the ocean, and they're affected by heat especially. Heat can break up the structure. It breaks up the folding. It doesn't mean that the protein is necessarily, it's lost its chain of amino acids, but just if it falls apart geometrically in folding, it's no longer effective. These proteins, I think for E. coli, the average protein lasts maybe, less than a day, maybe 20 minutes, I don't know. You have to have a continuous way of making these proteins. Having one or two proteins formed somewhere in the world is not going to help you any. That's not going to give you life. And finally, let's look at the odds here. We discussed this in some of my other tapes, some of the other videos, and look it up. If you have a simple necklace, let's say you have 4 pearls, 5 kinds of pearls, but this necklace only has 4 pearls, but you can choose from 5 different kinds of pearls. The number of combinations, and you remember there's only one right combination for each protein, and one right combination, more or less, for each DNA ring. The number of combinations is the number of types to the power of the number of positions. For a protein, we have 382 proteins in this simple little bacterial cell. And as we saw over here, unfortunately, if it started from basic chemicals, then instead of having just 20 amino acids, we now have 40, because we have both the left-handed ones and the mirror image right-handed ones. So instead of what we showed before in other videos, instead of 20 types, we have 40. The number of amino acids in an average bacterial protein is about 250, 300. Let's say it's 250. So it's 40 to the 250th power, which is a much, much larger number. Again, look at the other videos, than the 10 to the 80 atoms in the whole observable universe. That's for each of these proteins. There are 380 proteins that you have to form individually. So not only they're unique, they're not only separate, they're unique proteins. Each one is different. So you have to do this 382 times to look at the odds here. only one of these combinations, 40 to the 250 zeros, is the right combination for that protein. And DNA is even worse. DNA is composed of 4 nucleotides. For M. genitalium, the size, the DNA, is 580,000 pairs. So, to use the same formula here, you take the number of combinations as the number of types, which is 4, to the 580,000th, raised to the 580,000th power. What probability do you have of getting the right kind of DNA? Everything has to work together all at once. There is no way, from simple chemicals, no matter what you do, that you're ever going to get a living organism. Thank you very much.